want to take you through PCR, which is polymerase chain reaction. This is a technique in genetics that is talked about a lot in science classes. I think many students have no issue telling you what the 3 steps of PCR are, maybe even drawing a few pictures about that process. But much fewer students have a really firm understanding of what's going on at the chemical level of PCR and can adapt it to different situations. Okay? So this will serve as a primer for that ability. Okay? So first thing to understand is that there's 3 basic conditions in PCR, and it's driven by temperature changes. Okay? And those 3 temperature changes are as follows. Okay? First, we're going to start our reaction hot. We're going to drop the temperature to something that is relatively cold, and then we're going to settle in a nice medium temperature. Okay? So when you look at a PCR machine, all that is a fancy oven that's changing between these 3 temperatures on some specific time course, usually in the matter of seconds or minutes. Okay? What is in that mixture? Okay? PCR happens in a single tube, and there's a couple of different ingredients that are going to interact together based on these temperature changes to produce what we want to create, which is ultimately a PCR product. Okay? And the PCR product is a double-stranded piece of DNA, relatively short compared to a genome or something like that, but which consists of a particular target we were looking for. So each individual PCR reaction happens in a tube, okay? Usually in a very, very tiny tube with anywhere from 20 to 50 microliters of fluid, most of which is water. So here's what that tube might look like. Okay? One thing that we have to have is a template. Oftentimes the template is genomic DNA. Okay? But it could be any sort of extended piece of double-stranded DNA from which we want to isolate a smaller piece of that DNA. Okay? We have a template. We have a forward and reverse primer. Okay, I'll explain what those are in a second. Okay, you also need a DNA polymerase, okay, which is the protein enzyme that's going to drive the reaction to create the new DNA. Okay, you need free unincorporated deoxy nucleotide triphosphates. Okay, we refer to those as DNTPs. Okay, those serve as the building blocks for the reaction we're doing. Okay, we also need some other things, some salts to keep the reaction happy, some magnesium ions to help soak up some of the phosphates that are going to be lost as we add these nucleotide triphosphates. We give off 2 phosphates in that reaction that have sort of a net double negative charge, and that magnesium helps soak those up and keep the reaction moving. Okay, and then of course some water, okay, to keep all those ingredients swirling around and happy. Okay, other than that, those are going to be driven forward in the reaction by those temperature changes that I laid out before. Okay, so we have all our ingredients together in the tube, in the PCR machine, and it's time to start driving the reaction forward. In this case, I'm using a genomic DNA template, which is double-stranded. Okay? Think of this as going really far to the left and the right. Let's say this is a particular chromosome that may be millions of nucleotide pairs long. Okay? And in this reaction, we also have our forward and reverse primers. Our forward primer and our reverse primer are both single-stranded, short nucleotide stretches. Okay? Usually between 20 and 30 nucleotides long. Okay? The identity of all of our forward primers is exactly the same. There's millions and millions of copies of this forward primer in our mixture. The identity of the sequence, that 30 nucleotide base sequence, will determine where on our genomic target that this is going to base pair complementary to our target. Okay? So if this is a string of all C's, then this primer will seek out a string of all G's to complement to, and it will stay at that location only. It will not bind by hydrogen bonds to anywhere else on this target chromosome, because that sequence we've designed to only complement one particular sequence on here. If there are a number of internal mismatches on that primer, it will not bind effectively to that site. Thus, between using a forward primer of 20 to 30 nucleotides long and a reverse primer, which is also unique to a particular target, we can define an exact area by which these two primers will stick by hydrogen bonding to its target. Okay? To do that, though, we have to open up this double-stranded template with heat. Okay? So that first step in the PCR process, to heat the sample, usually to something like 94 degrees Celsius, okay, which is pretty darn hot, which is close to boiling, is enough to separate all the hydrogen bonds in this particular mixture. Okay? The number of forward and reverse primers greatly outnumbers that of the template DNA that's in that mixture. So the primers, which are smaller, much smaller, and more nimble in a chemical sense, will always reach their target before this big long piece of DNA is able to come back together. Okay? So we pick the sequence of these particular primers to lay down on a target which is right next to each other on this linear piece of double-stranded DNA. So for example, if we were to heat our sample, okay, that's going to separate these two strands, okay, making the DNA sequence on the top and the bottom exposed. So now we have exposed single-stranded DNA, which is our template. And this forward primer, if it so matches, can sit down on a particular target, hydrogen bonding to it, okay, when we lower the temperature. Okay? So that first step in PCR is to heat to 94 degrees Celsius, thus opening our target. We then very quickly cool, okay, to maybe a nice low, in chemical standards, low temperature of, say, 50 degrees Celsius. That allows the primers a selective advantage for that site, and they get here before the DNA closes back on itself. Okay? So that's where our forward primer will be. Notice that there's a 3' hydroxyl group at that tailing location. That will become important. Okay? And a reverse primer is going to bind, by design, to the opposite strand, sort of downstream from this location. And we've designed this in such a way that the 3' hydroxyl group on that primer will be oriented the opposite direction. In this way, we're defining this region of our template DNA via our two primers, such that when we amplify or we build DNA off of this template backbone, we're building into one another, sort of crossing over that region. Okay, so let's look at this at a little different angle. All right, so we've got our reaction that has our template DNA in it. Okay, we've heated the reaction to 94 degrees, which is going to separate that template into two single strands, allowing us access to all of the nucleotides that may be there. Okay, we're going to drop the temperature of the reaction to a relatively cold 50 degrees Celsius, which allows our primers forward and reverse to stick to their respective targets within that genome or that chromosome, to bind stably, and that provides inward-facing 3' hydroxyls. Okay? We say that the primers are flanking the target being this middle section here. So at that cold temperature, the forward primer and the reverse primer flank that central target left and right, and set up the possibility to build new DNA. I'm showing here in green, that direction on the top strand, okay, and this direction on the bottom strand. And you can see effectively what we've done is we've copied that target region in the middle. Okay? The DNA that's built here complements the top. It reflects that bottom template strand in black. Okay? And down here, this green strand, this growing strand, replicates the top template strand up here, okay, because it's complementary to that. Great, now let's look at the next step. Now, this third stage of the PCR reaction is referred to sometimes as elongation. Okay? Elongation is what's happening here in green. We're building new DNA. That's not happening by magic. Okay? We're raising the temperature back up to a medium level of about 72 degrees Celsius. And at this temperature, there are special DNA polymerases called TAC polymerases, TAQ polymerases, that are heat resistant, that like to build DNA at that exact temperature. Okay? So at 72 degrees Celsius, they're activated, and if there's a primer in place that has provided an open 3' hydroxyl group, and given that we have plenty of free, unincorporated deoxynucleotide triphosphates, which are those building blocks of DNA, those can be incorporated by that DNA polymerase, okay? Piece by piece, very quickly too. It only takes about 1 minute to replicate about 1,000 bases of DNA under these conditions. So if my target region here is, say, 2,000 bases long, in 2 minutes I can replicate that whole region anywhere there's a forward and reverse primer sticking. Okay? You can see that the outcome of that is we've taken one target piece of genomic DNA, okay, and we've made 2 out of it. Okay? In this way, if we start with, say, 10,000 copies of my template genomic DNA, after 1 round of PCR, which only takes a number of minutes, I'm up to 20,000 copies of that target. Only at that region, though. Keep in mind that my template DNA may go left and right of this particular diagram, millions of nucleotides. The only region that I'm actually copying occurs between the forward and reverse primer, and it doesn't include the rest of that template chromosome. Okay, what I've just described is one complete cycle of PCR with steps hot, cold, and medium temperatures in that little fancy oven that's driving this reaction. So when you've completed that, you've gone effectively from 1 copy to 2 copies of your particular target. Okay? And you can see that here. In the green, I'm showing the new DNA that's been created, and in the black, the original template DNA. Okay? All we have to do now is run another cycle of this reaction. All of our ingredients are still in the same mix, and we can continue from there. Okay? So let's answer the question, what's going to happen when I go back to hot? Okay? Under these hot conditions, my 2 new strands of DNA are going to separate themselves. Okay? So I'm going to get a scenario kind of like this. Okay? Where my old DNA is separated from my new DNA, and now I've got 4 templates instead of the original 2. Okay? My forward and reverse primers will again bind to their complementary sequences. This time they have more things to stick to. Okay, so I hope you can see that. That provides those 3' hydroxyl groups at those key locations again. Okay, then I'm going to, of course, that happens as a result of a cold temperature that I've put on this. When the temperature drops, those primers, which outnumber the template, find their spots really quickly, preventing those from closing in on themselves. Okay, and then we raise the temperature slightly to a medium level, which makes our DNA polymerase, also called a TAC polymerase, very happy. Okay, so our polymerase is happy. It's going to bind at those locations where the 3' hydroxyls are available, and it's going to create DNA copies extending to downstream of those 3' hydroxyls. And what you can see is now we have 1, 2, 3, 4, 5, 6, 7, 8 strands of DNA representing 4 copies of our original target, our original template. Okay? So after 2 cycles, I've gone from 1 copy to 4 copies. After 3 cycles, I will be moving to 8 copies. After 4 cycles, 16, and so on. What you're going to get is an accumulation of double-stranded pieces of DNA that represent the region between my forward and reverse primers. And we can very quickly, in 35 to 40 cycles, get that copy number up in the billions. Okay? That gives me this little double-stranded target, in the billions range, 1 times 10 to the 9th copies, which I can then use in other applications. I can study it a little bit easier because that signal has gotten really big, and it's gotten so big that it greatly outnumbers the genomic DNA, the large pieces of DNA that were there before, and I could use a DNA gel perhaps to separate out those smaller fragments and do something more specifically with those. That's a very useful tool in biology. To summarize, right, we've started with a long double-stranded DNA target with many, many different kinds of bases, but we've designed forward and reverse primers, these short single-stranded nucleotide stretches, to complement regions that are neighboring one of each other or flanking one another, is what you usually see written, such that if we change the temperatures in a fancy way, they sit down at those regions, and DNA polymerases extend the DNA between those regions, creating multiple copies of that. After one round of amplification, I've got 2 copies of my template. After we sit primers down again and extend the DNA, I've got 4 copies. After we sit primers down again and do it again, we've got 8 copies, and so on and so forth. We just keep creating the DNA within that region, and we end up with billions of copies of that particular target.